Ahora.
Okay. Sorry, guys. Too complicated nowadays. Things. Okay, so well, Miguel Alvarez is gonna tell us the progress he has done so the last month on his research for the PhD. Okay, and so he's gonna talk about the work going on the formation of coral reefs. So yeah.
Okay, thank you very much. So today I'm presenting the progress I've made during the first year and also the second. Perhaps we have to mute the other participants. Yeah. Okay. And yeah, so to start, well, this work is been doing in collaboration with my supervisors, Manuel Matias. I forgot the A between Manuel and Matias, and Damia Gomila. And but the good news is that more names are coming to this list because we have started a nice collaboration with the marine ecologists that will feed the model with empirical data, and also they are assessing us with the ecological interpretation of the model. So to start, why it's important to talk about corals nowadays? Basically because they are in danger. So coral reefs are quite an important ecosystem for the world. So this is an infographic I got from the Nature Conservancy, in which they display some very key data. For instance, that coral reefs perform quite an important labor on coastal protection, since they reduce the wave energy by 97% before hitting the shore. And there are some countries that are directly built upon our coral reefs, the case of the Maldives, for instance. Although they cover only 1% of Earth's surface, they host 25% of all marine life. And also they are a source of a lot of money, 36 billion through tourism, but also through food production. Since coral reefs, they host a lot of fishes, a lot of seafood, and they provide food for 1.2 billion people globally. So this is to make a little bit of introduction of why corals are important and why are they in danger. They are exposed to many threats nowadays. Some of them are, like, let's say, natural. This is the case of bleaching. This is the same coral colony in two stages. This is a healthy coral. This is a coral that has suffered bleaching. I don't know how much do you know about corals, but corals usually live in symbiosis with microalgae. And these microalgae provide corals with the vast majority of the nutrients they need. And when there is thermal stress, which is suddenly is quite typical nowadays, corals expel the microalgae. So all the color and the primary source of nutrients for corals abandons the coral, and corals go white. So these are not dead yet, but this is a clear route for death for corals. And this is increasing because of the warming of the waters. But also corals have predators, like the crown of thorn starfish that literally eats corals, and so do the parrot fishes. But also there are some human threats, like tourists, plastic pollution, dynamite fishing, and on top of all of that, global warming. And global warming is not only, let's say, bad for itself, but increases two of the main threats for corals, which are bleaching, that I already mentioned, but also ocean acidification. Since corals are the stony corals are made of calcium carbonate, the fact that the ocean acidifies means that corals literally dissolve or cannot produce strong rock structures to survive. So what are we going to do to save corals? We are going to do models. And to start with the models, we have to think a little bit of what are corals before starting with the maths. Corals are animals. I like to start with this because it's the first thing I learned during my thesis, that corals are animals of the phylum Cnidaria. That means that they are some cousins of jellyfishes. They are quite an old species. The first species of coral appeared 5 million years ago. Just to put in context, Australopithecus, the first hominid, appeared only 4 million years ago. As I already mentioned, they usually live in a very quite nice and strange symbiotic relation because it's completely unstable. This relation is very sensitive to change in temperature, very narrow change in temperature from 2 or 3 degrees put in danger this relation. And these corals obtain the majority of nutrients from this photosynthesis that occurs in the microalgae they host. So corals are primarily a clonal organism. You can see here, this is a colony of stony coral, a species of brain coral. So each of these cells is a polyp, it's a unit, let's say, of animal, and they assemble into colonies of the same species, and they reproduce mainly clonally, but also sexually. This is the life cycle of a coral. So it starts here with a healthy colony that spans gametes into the water. These gametes fertilize and span in an area of few or 100 kilometers. They travel down to the seafloor, and they settle there, and through clonal reproduction, they grow a new colony. So, as I mentioned, yeah, this slide is only to show some beautiful photos, but also to get some nice facts that corals assemble into colonies through clonal reproduction. There is a huge variety of species, shapes. For a same species, you can spot different shapes. And the most important fact, probably for the purpose of this talk, that there are some species of corals, stony corals, that synthesize aragonite, which is a phase of calcium carbonate, and use this aragonite to build an exoskeleton that is the origin of these beautiful structures. Polyps assemble into colonies, colonies assemble into reefs, which are these very nice ecosystems, and reefs grow through centuries. So when a colony dies, another coral colony can settle on top of the remains of other colony, and this structure grows and grows vertically and horizontally, forming very nice structures that can be easily spotted with Google Maps. And whenever wherever there are patterns, physics can do some work there. This is the case, for instance, of the coast of Saudi Arabia, in which you can spot reefs, coral reefs, forming parallel fringes. But perhaps the most iconic shape is the closed atoll that can be either empty or it can be filled with some inner structure. When I mean empty of coral, because the sediments of the erosion can form an island inside of an atoll. And you have this variety, these labyrinthic structures inside an atoll, but also in the more exposed edge of an atoll, you can see this, always I struggle with this image, but here there is a very nice periodic pattern of spurs and grooves. This is this part of the reef, which is the part most exposed to wave energy. And also at the big scale, you can see there is kind of some regularity and patterning in the disposition of different atolls. This is the Coral Sea in East North Australia. So the aim of my work is to understand or try to model how these patterns appear. And at the end of the talk, I will argue why this could be useful for the mission of saving corals. So what we did is to model this with a model of partial differential equations. Why? Short answer is what my supervisors do. Long answer, we have some reasons to do that. The first one is that there are some ecological mechanisms in corals that we know they are well reported in bibliography, and we know how to model those effects. For instance, you can see here in this plot the reduction of mortality of corals with the size of the colony. This is a clear effect of facilitation, and it's a hint that an ecological modeling, and we know how to do this kind of modeling may work. Perhaps more beautifully, interaction at different distances. It's a very nice plot in which what this plot is saying is that given a coral colony, the presence of another colony very close to that one enhances and has a positive feedback in the growth of coral. When you go to medium to far distances, there's a negative feedback that also shades with distance. Wherever there is long-range interactions, there is self-organization, and when there is self-organization, there is pattern formation, and partial differential equation models apply very well. So what do we expect for a model of these characteristics is that if we set a coral colony in top of a rock structure, you can see here, this is the rock structure that the reef has built, and this is live coral on top of it. If we let the time pass, we expect that the coral grows against the current, the main sense of the current, and grows both horizontally and vertically up to reach the sea level. There have been some previous attempts. Perhaps the two most honorable mentions is this one of Mr. and Bercovici, in which they model the corals as some kind of porous media that grows because the water currents are supplying the system with some resource that makes this medium grow. So they successfully reproduced the pattern of parallel fringes, and they vary in velocity, they vary the width of this structure, and also the spatial distance between these structures. Another nice model is this one in which they study the effects of the sea level rise and the previous topography. Now corals are settled in top of a slope, an inclined slope, and they study varying the diffusion coefficient of the carbonates dissolved in water, how different structures of reefs appear. But these are not enough for the purpose we want, so we build another new model, which, by the way, we claim is kind of minimal, although it's very complex. And so let's start with, to propose a partial differential equation model, we need a space in which we build the model. And well, this is a green sketch I prepared for my nephews to explain what I do, but then I couldn't recover the original file, but so you get this. So what we do is to parcellate the ocean floor, like this room, we parcellate in the square cells, and we consider whatever is happening in the water column, which is in the above these square cells. And we will define there some magnitudes that characterize the state of the system and propose time evolution equations for those magnitudes. So this is the network of eco, biophysical, geological interaction of the model. Here, there are many things occurring. I will break into parts, so perhaps you could understand it better. But just to say, there's a main agent, which is coral, the amount of biomass of coral. I will explain also what are the magnitudes I am working with. The amount of biomass of alive coral, that is the center of the model and has the most interactions with other resources, with other players of this game. For instance, the nutrients. Nutrients is like an umbrella word for resources or limited resources for corals. Carbonates, which are needed to build the exoskeleton of aragonite, which at the last dance we can treat as rock, hard rock. The variable rock and the height is easily converted into the height the coral has reached, because the more rock, the more height. And so for the purposes of the model, rock and reef height are completely equivalent. And also this rock modifies the ocean currents that are supplying with nutrients and carbonates the system. But let's break it into parts. First, define what are the magnitudes. There are some new definitions, others that we have recycled, but the main player, which is in this model, is P, is the ratio between the three-dimensional area of a live coral. So in this model, we treat coral like some kind of carpet, like two-dimensional structure that grows over the rock, over the ocean floor. So P is the ratio between the three-dimensional area of a live coral and the ocean floor area they cover. I don't know if you are familiar with this, but this is completely equivalent in the science of forest to the leaf area index. So it's a dimensional measure for the amount of biomass of coral. We also have A, which is the density of aragonite in the water column. As I said, this variable takes into account how much rock corals have generated, and is easily translated into the height the coral reef has reached from the ocean floor. You also have nutrients. As I said, this work doesn't, ecologists does not like nutrients because corals usually live in oligotrophic waters, but you can replace nutrients by limited resource and everything will be fine. We choose chlorophyll as a proxy for nutrients or these resources because as a first approximation, but the specific name of this doesn't matter really. And also omega, which is the aragonite saturation state. This is quite an important measure that is being used widely in the coral community because measures how easy it is for corals to synthesize aragonite. And it's quite it's heavily affected by ocean acidification, since the concentration of carbonates goes here. So I generally call, instead of aragonite saturation state, I usually name it as generally carbonates. And what is the interplay between all these four variables? First, the model is some kind of population dynamics in a wide sense. So what we will have for the main player, which is the alive coral, is some ratio of growth or death, depends if it is positive or negative, and also it's proportional always to the current amount of biomass of coral. So to start, the most obvious interaction is that corals uptake nutrients from the water currents. So there's a positive term with corals and nutrients, and appears with a negative term and a conversion factor in the equation of nutrients. So whatever mechanism is making corals grow requires nutrients, and always will appear with a negative sign and a conversion factor in the equation for nutrients. So what is the next obvious step is a death rate, which does not depend on the amount of nutrients. Here goes predation, temperature, human action, whatever you want. This is a mortality rate, and we put inside the model. More things going on. These ecological mechanisms, as I mentioned at the beginning, facilitation. Here we treat not facilitation as a reduction on the death rate, but an increasing on the growing rate. This is because, well, many things are going on here, but also the larger amount of corals means that they get more nutrients. So it appears here with a positive sign. But also we need, or we know there is some kind of interspecific, at the end of the day, saturation effects for corals. Corals cannot grow infinitely, so it appears a higher order term with P, which is competition between corals. And this term also prevents the corals to explode. So it maintains the amount of coral in a finite value. More things going on here. Corals synthesize aragonite. So they remove carbonates from the water column and transform those into rock. And they appear here in the same spirit. Wherever there are polyps and the aragonite saturation state is above 1, corals produce rock. And the same term appears with or without a conversion factor in the equation from the carbonates. These are all, like say, the building effects, but also the fact that the coral grows vertically and makes the alive coral to reach the sea level has new effects. The first one, sorry, I forgot that also there is an erosion effect on the rock that grows with the height of the reef, because the higher the reef is, the more exposed it is to the wave energy. Effects from the rock through to the coral. The nearer you are to the sea surface, the more sunlight you have. So it appears here with a positive term because it enhances the growth of coral, but also coral cannot live above the sea level. So we have included a saturation effect with the height of the coral. Here they are not displayed, but there are also effects, let's say non-local effects. Here appear spatial derivatives. This term reproduces the clonal growth. So when you put a structure of coral in your system, and this term makes this structure to grow horizontally. And also diffusion effects. And we put this here with two purposes in mind. The first one is to reproduce some kind of migration, fission that is reported in the literature, but also this term ensures numerical instability when you integrate the code. And finally, ocean currents supply whatever nutrients and carbonates corals are depleting from the system. So these ocean currents supply the system with an advection term and a diffusion term, and also the carbonates, which you see here, this completely an equivalent structure. So this is let's say, the overall model. But in top of that, we are lacking something, that is the rock modifies the ocean currents. And this link is missing here in these equations. And so we had to solve, this is new data. This is new results that I had in the last year. So what we have done to model how the presence of a coral reef modifies the ocean currents, we solved Navier-Stokes equations, not the $1 million ones, but a simpler version. Why, or the assumptions we have made to this point could make an entire talk on itself. but I will briefly talk about what levels of approximation we did, and how do we solve Navier-Stokes equations for our purposes. Since we are working only with 2 dimensions, I want to stress that the coral height is a variable of our model. We only work with a 2-dimensional field of a velocity, which depends on distance. This is an example of how of the solutions of the Navier-Stokes equations in my model. So basically, these are the Navier-Stokes equations: the explicit variation with time, the convective term, the pressure gradient. Since we do not care about the specific numbers of pressure, and we assume water is most, to the best of my knowledge, at this level is incompressible, and we replace by a general pressure phi, and also diffusion effects. And all this, and the Navier-Stokes equations for the 2 components of the velocity have to be solved along with the continuity equation. This may seem obvious, but we spent months working and failing because we did not take into account that we have to ensure continuity. So how did we solve this? Well, first we rearranged the equation, so everything is the same, but we have left the explicit time variation at the right side. And the idea here is that we didn't want to complicate a lot. Sea dynamics are very complicated. They are science on itself. And for the purposes of the model, we only wanted like an average effect of the currents on the coral reef. So we assume like if you imagine the velocity field of water as a kind of Fourier series in terms of time oscillations and spatial oscillations, we consider that like the basal, the zero mode, the one that does not depend, let's say, on variations and time in time, is the one that represents some kind of average effect of the current. So we simply replace this derivative by 0, and we can solve this very nice because now we have only dependence, or we have only spatial derivatives, and we can apply a method that we think we came with this method for the first time to solve Navier-Stokes equations. I didn't find this in the bibliography. It's not anything very nice, and it's not the very best method, but it goes with the spirit of let's keep it simple and doable for the purposes of the model, so I do not need extra packages or extra models. So the idea is to use Newton-Raphson method. I don't know if you remember Newton-Raphson method from computational physics at the university, but it is used to solve nonlinear equations and can be extended to solve nonlinear functionals, especially, and this is particularly easy if you are working with finite differences, as we are doing. So you might recognize what we want is to find with at the end of the day, an iterative method in which we update our variables. We want to find the solution that makes these expressions to be as closer to 0 as we want. So you might recognize that these 2 first functionals are indeed the Navier-Stokes equations, and this third is not exactly the continuity equation, but it will take, as I mentioned, I want to spend, I do not want to spend a lot of time on this, but this equation ensures, this equation for the pressure field ensures that the continuity equation is fulfilled. And now just to say, the idea of using this equation instead of this one is numerical instability, and also that here the pressure has also a second derivative that makes everything easier for the numerical purposes. So yeah, a lot of technical details, I will not enter there, just to show that this works. These are the X component and Y component of the velocity field. Here flux is entering from the right, so the blue means in this plot that the flux is traveling to the left, and when it goes to warmer colors mean it is reducing velocity. And here's the Y component. So the idea is the flux is entering from the right, and here you can see it goes up to surpass an obstacle. This obstacle is a tilted barrier in which we applied non-penetration conditions, by the way. And here the flux is going down to surpass the barrier. And you can see also there is a wake. So here the velocity is very slow. And we, as a gift, we have also the pressure field and the check we check that the continuity equation, this is mostly an almost homogeneous zero in the system. So we are pretty confident the model works for our purposes. And finally, we can meet all together. We have the model, we have the Navier-Stokes resolution method, and we have a table of parameters. Again, another talk of how they came up with this table of 13 parameters. But some of them are taken from the bibliography, some others have been estimated, and these parameters are in a constant process of updating and improving. So we have finished with the theory, we can get to the results. First obvious result we got is that there is a time scale separation. So usually corals have a very slow growing rate, so at the rate of centimeters per year, but water currents have a typical velocity of centimeters per second. So you can see here in this plot you have in red the distribution, the typical distributions of a live coral and rock the model produces. Flux is coming from the right, and in this plot you have how an initial flat distribution in this very light green of nutrients evolves with time. So darker greens mean farther times. So 0 seconds, 10 seconds, and after, if you wait for enough time, you finally get to this stationary distribution of nutrients. So I repeat, I put in the system this distribution of a live coral and rock, a flux, an initial flat distribution of nutrients, water flux is coming from the right, and the system naturally evolves to a state in which behind the coral reef there is less nutrients than in front of the coral reef. And in this plot, you can see kind of a measure of how fast the system goes from a flat distribution of nutrients to the stationary distribution of nutrients. Why this is useful? Because you can see here that this happens mostly in, well, approximately in 1 hour, while the average time step for a live coral is 10 years. So there is a clear time scale separation, and by the way, it's consistent with experiments in closed system reported in the bibliography. So there is a clear separation between the, let's say, the dynamics of nutrients and carbonates, which are being affected by seawater. I mentioned these different in characteristic velocities. So we, for the purposes of the model, or let's say if we are looking or interested in the live coral, we can treat those as stationary fields. And this is very nice because this simplifies the calculations a lot. And they are, if you work with finite differences, these equations are linear on the variables, and everything is very nice. You can make use of libraries of sparse algebra, and everything works fine. So more interesting results, finally, two-dimensional results of the model. What you are seeing here, I will again break this panel into parts, but this is the summary of what happens if we set an initial seed of coral of about a Gaussian seed of about, if I remember correctly, a few meters wide, and you let the time pass 15,000 years. This is what you got with the model after all this time. So some structures have appeared, and I will explain that in detail. So here in this panel, you have the distribution of a live coral, X coordinate, Y coordinate. This is the distribution of a live coral and the distribution of the rock left behind by corals. And this image is just for the purpose of, for the sake of comparison. The good news is that these results resemble what we observe in nature. This inside, all this inside is hard rock, and coral is lying in the outer edge of the reef. And there are some interesting features. If you make a cross section of these values, you obtain like very obvious results, but they were not reported in previous models. For instance, you have more distribution. This in red is the distribution of a live coral. In gray, you have the distribution of rock generated by coral. So here, the first obvious result is there is more live coral in the front part of the reef than in the back part of the reef. By the way, I have not a movie here, but this structure is traveling to the right, and the currents are coming from the right to the left. And this is the rock the coral has left behind. That resembles quite much atolls, and these are very good news, but also we have some extra information. This is the resolution of the of the Navier-Stokes equations around the atoll that produce distributions of nutrients of aragonite. Marine ecologists are seem very, I don't get the word, but they were very happy when they saw these results, because these are indicating this is the first step to begin to understand how different atolls can self-organize in space, since here there is a lack of nutrients and you have to wait for, let's say, 3 kilometers to find another big coral reef that has plenty of nutrients again. These are very nice results. We are working on this. Still, we have some work to do. But the good news is also that a reduction of the model, this was a 2D model, the reduction of the model to a 1D model in which we assume there is no Navier-Stokes equation, there is constant velocity, but there is advection of nutrients and all the other mechanisms. This produces different bifurcation diagram that resembles all the shapes, or let's say the most significant shapes of corals we spot in nature. So here you can see, I will show you again what are these different regions, but varying the income velocity of water currents and the ratio mortality-natality, you get different shapes of corals. So 4 basically means death because it's too much mortality or too low velocity, so no new nutrients are coming to the system. And the first one, this first region, what produces in the system is a single peak of coral traveling to the right. Again, water flux is coming from the right to the left and leaving behind this structure of rock that it resembles the structures of fringing reefs. Here, you only have a live coral in this part of the reef, and water fluxes are primarily coming from the left. So this is a regime. Changing a little bit the parameters, we can have 2 peaks in the system of, in this case, blue is live coral, orange is the rock. A peak traveling to the right, a peak traveling to the left, and this is you have to understand these plots as cross sections of a 2D model. So at the end of the day, what this represents is what I already showed to you before. These are 2 different parameterizations. We are still working to make these 2 plots fit together in the same framework. But whatever, what this plot is saying is that in this region of parameters, you have 2 peaks, so 2 regions of coral, one wider, one narrower, that represents closed atolls. And finally, if you move to very low mortalities and very high velocities, you have a lot of peaks coming, emerging from the inside of coral that somehow reminds these labyrinthic structures. This is a live coral, by the way, that appear inside closed atolls. And let's say we are happy. Yeah, we can discuss all these regimes, but the good news is that we can discuss this making use of a bifurcation theory. Perhaps this is too hard or too boring for some of the people in the audience, but what we have done, and we are working with this, we have reduced the model, another reduction of the model, to one that we do not take into account space, so the amount of nutrients and carbon is homogeneous. But the good news is that we can obtain a bifurcation map of the change in stability and behavior of the solutions, the stationary solutions of this model. So basically, I will not again enter in the details of what are all these bifurcations, but the point is that this line here and some things happening in this region can be used to explain why these different behaviors appear, because we can understand the different behaviors I showed you before as excitability. So here in this region, we have plenty of peaks originating. And if you move in this region at the right of this blue line, you have different types of excitability in the system, 2 peaks, 1 peak, or the death of the system. And just to arrive to the conclusions, what are the conclusions or the key points to the take-home messages of this work is that reef formation, which is a macroscopic phenomena, can be understood as the consequence of micro-mesoscopic interactions, this network of interaction I showed you before. And this is a very good news, or this is, let's say, this is challenging the most accepted vision on the coral community, which is Darwin's theory. So there is no need of previous structures or geological phenomena. Darwin's theory of formation for coral reefs, which, by the way, Darwin formulated this theory before seeing any kind of coral reef, says that there is an island, a volcano island, for instance, reef grows around this island, and because of some earthquake or geological phenomena, this island shrinks, and what remains is the atoll. This applies to some coral reefs, but it does not apply to all coral reefs. So this theory provides a new framework to understand reef formation from scratch, from microscopic and mesoscopic interactions. You do not need to invoke geological phenomena. Also, that coral reef formation in this framework are understood as pulses propagating, and since we have a whole science of excitability, this science provides us with a lot of answers to the question of why these different shapes. The interplay of few parameters is enough to reproduce these rebalance shapes, and the model seems to be useful. This means that some marine ecologists that we exposed this work to them said that these results may inform coral restoration projects, since they are reproducing, let's say, the natural course of events, and this is a hint or a help of where you should place new corals so they survive and repopulate the reef. And that was all. Thank you very much for your attention.
Well, thank you very much, Miguel. Well, it's time for questions. If there is any. Please, go ahead. Yeah, thank you.
Thank you very much. It was very nice. So my first question may be not relevant to you as a mathematician, but you said that corals are very sensitive to temperature changes. Mm-hmm. That's why climate change is posing danger to them. Not to sound like a climate change denier or anything, but there have been previous changes in the Earth's climate, and the timescale you presented for corals, I mean, they must have catched a few of those, and they still survived. So
yeah, you said it. There are time scales to the point that this warming is being very fast. I was attending a conference last week, and they were saying that corals probably will survive again. Probably they will be different as we know them by now. And by the way, there are some reports that in the Great Barrier Reef, they are being recovering quite fast from. But of course, we will have to wait to see what happens with corals. There is clear hope that coral reefs will remain, but we don't know in which shape. And the problem is that everything is changing very fast.
So say the last year on the 8 or something like that. So the change in temperature probably happened to centuries, no, something like that. Yeah. So probably through evolution and mutations, the corals adapted naturally. And now you change that in 50 years or something like that.
Yep.
Or it will be completely probably the problem is that what's not happening that it's going to get replaced by basic or something like that. So can we change completely?
By the way, there are being like ethic problems because there are projects of restoration using like super corals synthesized in labs, and some scientists are arguing that these is like a synthetic and artificial ecosystem, the one that they are wanting to reproduce. And by the way, the ones that are deciding which species are going to be settled on the corals are politicians, no scientists. So there's a lot of debate. It's hot, this topic in the area.
Okay. My other question is to do with the equations you are solving. So I don't know if you could maybe turn to the slide.
Yeah, for instance.
For instance, that one. So for example, you're saying that A represents the density of aragonites in the water column. Okay, so that's like the kind of rock that has been formed. So my question here is that when you're doing these kind of equations and dynamics, for example, for the most well-known of these are predator-prey models, right? And you can say, okay, so it's predator times prey because it's a probability of encounter. But here you do have various dimensions in play, right? Because you're taking, you have the water current, which is affecting this column of coral only in its cross section. Then you also have, the coral that is a 3D living structure, but it's only, growing on by its surface area, right? Because it's leaving the hand. So you have like a lot of I don't know, scales and dimensions there. So, okay, why in particular do you choose, for example, that, that minus LA over there? Why is it a to the power of 1 and not to the power of 2? How do you, kind of your intuition and why you choose this particular form?
That's a very good question. And I navigated through the bibliography, in particular, in that case, in the erosion rate, and you don't have a lot of information of, there are plenty of measures of, for instance, the erosion effects. And there is no clear, or I didn't found any clear law of growing with height. So this is something I put there on my own. And the short answer is that equations must be, the time evolution must be 0 when the coral reaches the seafloor. So you have to have a proportion with A, otherwise you could have negative concentrations of aragonite. But yeah, the answer will be that this is the simplest one we came at. And concerning the amount of scales, yeah, this model, by the way, the first big approximation of this model is to compress all the 3D dynamics into 2 dimensions. We are working only here with X and Y. We are locating our cells in a 2-dimensional space, and height is a variable of the model. So why did we choose these ones? Many reasons. For simplicity, they have previously used in the literature that you say predator times prey, which seems to be the most natural and the most why I will put there another power. Yeah, let's keep it linear, since we don't know almost anything. The reality is that this is completely wrong. By the way, I'm thinking how to improve this, but many let's say non-trivial functions, sigmoids, step functions, exponentials, they could go in there, but I decided to keep it, let's say, the most simple I could. And by the way, you see, this is not simple at all.
Like my point of view on this piece. So that's certainly going to be a complicated function. So the temporal drift of A will be a complex function, complicated function of A and B and everything. And we are trying to capture the minimums in variance here. No? So as I see, the usual thing to do here is that when you have a function that you know what that is, what you do is a Taylor's function of that, and that's the case. And then you have a reason to say that that's 0, that should not be 0, so that's the one you pick, and you check what the corridor is. And you can see from the measurable in some way. So that's the way we proceed somehow. So we are keeping the first term of a Taylor's function of non-linear function that we don't know. Okay, so if you don't have any other reason to put a larger than 1 exponent there, principally, that will not be that.
Right, but well, the thing in particular, I understand. I mean, that's usually what one would think. You say, okay, it's Taylor expanded, but I mean, maybe in this case, if you're having meters upon meters of rock compared to the other scales that you have over there, which is 1 to 100 centimeters, and then you show me results across various kilometers. I mean, you're maybe far from that Taylor expansion range, but of course, I mean, minimal model, yeah.
By the way, I saw in many papers, especially the ones concerning the nutrient uptake and the hydrodynamic forcing, these are filled with power laws, with non-trivial exponents. So you are right. You are completely right. This, but the point is that I didn't want to. Yeah, in the spirit of what Damia said, I don't know what these functions will be. I don't want to imagine them as very complicated functions in a first step. But you are completely right. You are completely right.
And also about the parameters, where do you get all of these parameters? Was it easy to find? Are some directly measurable, or how do you work your way around that?
There are some that are clear. I took from the bibliography. For instance, the diffusivity of species dissolved in water, the amount of chlorophyll dissolved in water, the saturation state. This is very well measured nowadays, and many of the others. For instance, mortality. Let's say basal mortality. You put there like the order, more or less the order of the lifespan of a coral. You can have a plus minus the predation effect, but more or less you have many, but other ones have been how would say tuned or estimated better. They have been estimated, so the model reproduce what we observe, or the time and spatial scales fit with the model. And since some of these parameters are come from interactions that do not go in this way. So coral dependence with height do not go as a parabola. Goes with an exponential that cuts when the coral reaches sea level. So for instance, this parameter G has no ecological sense in the, let's say, very strong ecological sense, the specific value, I mean. This is a mess. I'm constantly working on improving this table and trying to make this table as close to the reality and bibliography. And as I mentioned at the beginning of the talk, the team of marine ecologists that has joined this work will help with this. And during this summer, they're going to do an extensive and intensive exploration of bibliography to improve these parameters. The good message is that in this model, at the end of the day, what changes the dynamics are combinations, dimensional combinations of parameters. And as long as you keep those in more or less in the same values, the results of the model will be the same.
And for the time scales, for example, on the second one, when you approximated derivative to 0, can you go to that slide? Right. So which one is what you are putting to 0? Because you're saying here that the characteristic velocity of the things that you're setting to 0 is actually the highest one, right?
Yeah. So because they get more rapidly to the stationary state. So the dynamics, they push these variables to the stationary state quite fast.
Okay.
Very well. More comments or questions? Okay, why not?
I had a question about the clonal term that you put, because I don't know if what you modeling is the clonal reproduction inside one clone, no? But the typical scale of one clone of coral, I don't have the idea how much big, but I imagine like this. So from a macroscopic point of view, I don't know what is the role of this term. No? I mean that a clone that is in the scale where the clone reproduction plays a role is small in comparison with the scales you are considering.
Yeah,
here.
it's very small, but it is the way it spreads. So at the end of the day, this produces a lateral expansion of whatever distribution you have.
But how big is a clone of coral?
Centimeters.
A clone, sorry, like all a clonal. I mean,
So what's cloning is the polyps.
But how big is yes, maybe a clonal domain. I don't know. The individual. Yes, the structure of clonal polyps is usually something like this. No, it's just a coral. Notice the full reef, or
Yeah. But we are considering that the full reef is clonal at the end of the day. That, and by the way, we are considering like coral reefs as a single species, and there are plenty of species. So this is kind of a mixture of every building reef coral. Yeah. Also, all the reef is clonating in the direction of marked by the clonal term. Yeah.
The point is that there are many species in a reef.
yes, I understand.
The name below, all of them is clonal.
Yes, but
So not sexual reproduction.
as I imagine it, is like you have one clone, it grows up making this coral, but then it stops
I got
growing
your
at
question.
some point, and to grow, no? Don't. Okay. Maybe I have that in mind. Definitely, like seagrasses or okay. I can understand.
Yeah, yes and no. When you see photos of corals, what you have usually is finite colonies.
Yeah.
This is what you mean, right? Yeah. But at the end of the day, you have many of these that and the new ones, the new sexual ones that that settle in the seafloor have more chances to survive the closer they are to the previous reef. So the overall effect is when a new colony settles next to one you have, is like they have reproduced clonally. So if one of these appears next to this one, because a larvae creates a new colony of this,
Okay.
the overall effect is that they have reproduced clonally.
Okay, so it's not clonal, but
Yeah. Of
Okay.
course, each of these ones
okay.
generally is the origin of these ones is sexual,
Okay.
but the larvae of coral have more chances to survive the near they are
Okay,
to
okay.
their
So
the
it's
actual reef.
sexual, but very close.
Very
to the-
close.
Okay.
Yeah, there are also effects of long. These are not included in model, and sometimes we think about including these ones, but the fact that larvae are transported by Lagrangian transport through the ocean is what originates different reefs at the end of the day, not the fraction of a big one.
Okay.
Also that effect is that we do diffusion that we have to some application. So you have also more diffusion. So you have also more.
Okay.
Typically, we cannot dissect the production and saying that typically more or less a diffusion effect. That also to actually work. And that goes to that both are giving the function of the
Yeah. Thank you.
Thank you.
Thank you for your presentation. So let me preface this by saying that I know nothing about corals, but I'm interested in the structures behind you. That is the shapes of the different colonies. And I'm wondering how much is known about the process whereby these are formed. Is it primarily driven by genetics of the individual species, or is it driven by physics or some combination of that?
There are many things going on here. In fact, there is a physicist, Merckx, which works in the Netherlands. He has done a complete work on why these shapes. There is a genetic component because not every species, for instance, that one in the right, does not produce like these tree-like structures. They're very ramificated. They remain like the name of those types of corals is brain corals. But for instance, one of the most famous species of corals, Acropora, which is this one, they tune the shape through and they adapt to physical, to environmental factors. For instance, the light availability, hydrodynamics, the place in the reef in which they are settled. Because if you are very close to the sea level and you have a very spread shape, the first wave that comes in breaks the reef into parts, and the table corals are usually in the lower parts of the reef, and these ones are very compact are in the top part of the reef, and then those very branching corals are in the middle part. So genetics play a role also, because there's a major distinction between octocorals that spread into 8 clones, or the most common type of coral is spreading, say, 6 clones. It depends if you are a soft coral that you not synthesize an hard exoskeleton, or if you are a stony coral that you synthesize a hard exoskeleton. So it is driven by genetics, but the same species has a lot of, let's say, versatility and various shapes depending on physical factors. And there are works. Indeed, a scientist, Eva, is working on a microscopic model of why these different shapes with a minimal set of parameters that represent light availability, hydrodynamic drag, these things.
Eva is one of the postdocs. She's working on the microscopic models of and she has a nice model with just one, so you have elongation and branching, depending on the probability of elongation and the angles. You can reproduce many of the shapes with one parameter, or I think just one.
Two, three. Mm-hmm.
So yeah, it's a nice problem.
But usually, I don't know if your question goes in that direction. They are done with agent-based models. So I have not seen a lot of, let's say, PDE models, pattern formation in the classical sense of Turing stabilities concerning these specific shapes. That would be a very nice work, by the way.
I think your case has a model. Don't remember what cellular growth or something like that. You have this branch also.
Yeah, I mean, there are many models nowadays of growth of multicellular structures, organs, and so that type. And some of them require some physical constraints, internal stresses or whatever, could be temperature of the water or something. And if you vary those, you get different kind of structure. So I'm just wondering whether that also occurs here.
It's certainly okay. I think we stop here. We thank Miguel again, and thank you.
Next.